floating respiration. That is floating respiration. Now, the burning of protein is called cytoplasmic respiration, right? Is that clear to you, Rabia?
Yes, sir.
One very important, one more important. Suppose that there is a person, he or she is going for hunger strike. First, what will happen? Now, he or she is not taking any kind of the food because she is in the or he is in the hunger strike. So that person, what will happen? First, complete carbohydrate of the body will get burned. So utilization of the carbohydrate will take place. Then what will happen? After when the complete carbohydrate will exhaust, now it is the number, now it comes to the fat, right? Fat of body, it will start burning. Now because there is no carbohydrate. Now, and the fresh carbohydrate is not being supplied to that person. Now what will happen? Fat will start burning. If the person is very healthy, then he or she will have more fat. He or she can spend more time without dying, right? If the person is skinny or it's very thin or slim, the person will die easily because he or she may not have that much fat, right? Then after the fat, when complete fat will burn, what will happen? Now it comes to the protein. Protein is not respiratory surface. Now what will happen? Body will start, our cell will start eating itself, because our body is made up of protein, muscles are made up of protein, skin is made up of protein, right? So most of the body structure is made up of the protein. Now the cells will start eating itself and the person will die. That's why it is called cytoplasmic respiration, because the mitochondria will start eating the cytoplasmic structure. That's why this is also called autophagy, right? Cell will start eating on destroying itself. This is called autophagy, and the person will die, right? Is that clear?
Yes, sir.
Please note down. When carbohydrate or fat, when carbohydrate or fat is used as respiratory substrate, when carbohydrate or fat is used as respiratory substrate, we call it floating respiration. We call it floating respiration. Full stop. Normally, next point. Normally, protein is not respiratory substrate. Normally, protein is not respiratory substrate. But when protein is used as substrate, this is called floating respiration. This is called floating respiration. This floating respiration question I've been asked, right? Now all these respiratory substrate are converted into acetyl coenzyme A or organic acid. This acetyl coenzyme A or organic acid is used in Krebs cycle. It gets burned, and production of ATP takes place. So energy is produced in the form of ATP, adenosine triphosphate. Why? Because this is adenosine sugar. This is adenosine. Pentose. It's a nitrogen base basically. So adenosine, and there is a 3-phosphate. These kind of the bond, these sigma type of the bond, they show that they are the brokeable bond. So what happens when they are weak bond? So and they can be easily broken. When energy is required, ATP will break down into ADP, adenosine diphosphate. and one inorganic phosphate, that is PI, is released and energy is released. Again, in more emergency situations, right? If there is a more energy is required and ATP is not there, ADP will break down into AMP, that is called adenosine monophosphate. One more bond will break down, and further the energy will be released. So that's how the ATP is converted to ADP, and then ADP is broken down to AMP. So again, AMP and ADP are converted into ATP. So this cycle is keep on going. Is that clear, Rabia? Any doubt? All clear?
No, sir, no doubt. It's clear.
Fine. Now the type of respiration, right? So it's easy. So basically, you must have seen types of respiration is easily, but how much energy is released, that is the question. And it was not an easy question. I have seen the question in the previous year question paper. So respiration is basically of two types, aerobic and anaerobic. Aerobic, where oxygen is needed for burning, and anaerobic, where oxygen is not needed for burning. So in aerobic respiration, burning of food or oxidation of food takes place in presence of oxygen, right? So normally in normal condition, aerobic respiration takes place in plant body and animal. Whenever we talk about the respiratory substrate, so normal or most preferably the respiratory substrate is glucose. Glucose break down in the presence of oxygen. It is getting oxidized. 6 carbon dioxide is removed, 6 water molecule are removed, and 686 kilocalorie energy is released by the breakdown of 1 glucose. In anaerobic respiration, it takes place in absence of food, where the burning takes place but oxygen is not present. Anaerobic respiration takes place in bacteria, fungus, and human muscles during the hard work or running. So in our body, when there is no sufficient oxygen supply, or the supply is not proper, oxygen supply is not proper, it takes place when we keep on running. It takes place when we do the hard work. So in those situations, aerobic respiration does not take place. Anaerobic respiration takes. So anaerobic respiration in bacteria and fungus, it takes place. C6H12O6, glucose is respiratory substrate, it gets burned. Carbon dioxide is released, energy is released, but water is not released. In place of water, formation of ethyl alcohol take place, right? Is that clear? Now the energy is released only 59 kilocalorie. That's why we can call this kind of the respiration is inefficient. It's not efficient like aerobic respiration, because the bacteria and fungi are the lower organism. They are not that much evolved like the animals. That's why in lower organism, this takes place. And this kind of the respiration is also known as what we call this fermentation, right? It is called fermentation. Clear? Again, but one more important. When anaerobic respiration takes place in our body, on those places, formation of ethyl alcohol do not take place. In our body, what happened during the fatigue, during the hard work, this carbohydrate break down in the presence of enzyme into lactic acid, not in ethyl alcohol. So when the anaerobic respiration takes place in human body or any animal body, on that situation, the formation of in that situation, the formation of ethyl alcohol need not take place. Formation of lactic acid will take place, and only 36 kilocalorie energy released. Right now, what happened? So suppose that try to understand what exactly I want to say. Suppose that I have started running. So first, what will happen? My muscles of my thigh, legs, and hand, they require more energy. So they will start burning. Now they will require, for the burning, they require more oxygen. But lungs are taking, like they are breathing in, breathing out frequently, but they have their capacity. Lungs are working at its full capacity. Now the oxygen is supplied with the help of heart. Heart is pumping at its full capacity. Still, they are unable to provide that much oxygen which is needed by my leg. So what will happen? Some of the muscles which will not get the oxygen, they will start anaerobic respiration. Then Due to this anaerobic respiration, what will happen? Deposition of lactic acid takes place in my muscle. That will call muscle cramp. I will feel pain, muscle cramp, and I will sit down somewhere. This lactic acid, this is the point which you need to understand. Please listen carefully. This lactic acid will get deposited in my muscles. When I will feel the muscular cramp. Now I cannot run more. Then what will happen? I will sit down somewhere. Now this lactic acid, deposited lactic acid, it will go to where it will go. It will go to the liver, and in liver this lactic acid will break down, right? And I will feel comfortable, and my fatigue will be. Is that clear?
Yes, sir.
Please note down a point. In human muscle, anaerobic respiration in human muscles, anaerobic respiration takes place in human muscles. Anaerobic respiration takes place during hard work or running. During hard work or running. Full stop. Formation of lactic acid takes place. Formation of lactic acid takes place in this process. Formation of lactic acid takes place in this process. Full stop. Lactic acid, this lactic acid gets deposited. This lactic acid get deposited in our muscles. Full stop. From muscles, it is transported to liver. Underline, transported to liver. From muscle, it is transported to liver, where it breaks down. Full stop. So this was all about the aerobic and anaerobic respiration. Now, what is the respiratory quotient? RQ have been asked number of times, and this question was directly from the NCERT. So what is the RQ? Respiratory quotient, right? So the respiratory quotient is volume of oxygen evolved, like this volume of oxygen. Volume of carbon dioxide evolved, this carbon dioxide, and volume of oxygen absorbed or means required during the process of respiration. So that ratio is called respiratory quotient. Very important thing I want to share with you. Please note it down. The substrate having more respiratory quotient, the substrate having high respiratory quotient is the better substrate for respiration. RQ is 1 for glucose and carbohydrate. Rabia, you need to cram it. Anyhow, you have to remember it. Number of the time it have been asked, the respiratory quotient for the fat is this option is there. So you have to answer accordingly. RQ for glucose or carbohydrate is 1. RQ for organic acid is more than 1. That's why this is best respiratory substrate, right? RQ for the fat is 0.7. It means the fat is the worst respiratory substrate. RQ for protein is 0.9. It is also a good respiratory. What is the compensation point? I have told you what is the compensation point, but I will repeat again over here. And I ask you to please note it. What happens? Suppose that photosynthesis is taking, right, in any cell. Because of photosynthesis, oxygen is produced. Oxygen is utilized in respiration. Because of the respiration, this carbon dioxide is released. This complete carbon dioxide is utilized by the photosynthesis. And again, this oxygen completely utilized by the respiration. So the carbon dioxide, neither it will take carbon dioxide inside, nor it will release oxygen. So that photosynthetic cell will neither take carbon dioxide nor release the oxygen, right? So exchange of gases will not take place. Net exchange will be zero. This point is called compensation. Is it clear, Rabia?
Yes, sir.
Please note it. Compensation point in a photosynthetic cell is that point at which net exchange of O2 and CO2 is zero. Net exchange of O2 and CO2 is zero. Full stop. The complete oxygen produced during photosynthesis will be utilized by respiration. Full stop. The complete CO2 produced during respiration will be utilized in photosynthesis. Now, Rabia, try to understand this, right? You will not find this diagram anywhere. This is the complete summary of the respiration. How the respiration takes place, where the respiration. So I want next 5 minutes your concentration, 100% concentration. It will make you understanding of respiration, 100% understanding of it. So look, I said whatever we eat, like carbohydrate, that is carbo. So mostly we eat bread, we eat wheat, we eat rice, we eat potato, we eat pulses. They all are made up of the carbohydrate. That is a starch. So in the nutshell, maximum sugar what we eat, that is a starch. Starch goes inside our stomach, our small intestine, and finally it is broken down with the help of enzyme into glucose. This glucose is absorbed by our small intestine through villi. Now the glucose goes into our blood. Now the blood distributes this glucose across the body and transfer or provide every cell glucose. Glucose is taken by the cells. Now glucose is capable enough to go inside the cell with the help of plasma membrane and pores. Now when the glucose will reach in the cytoplasm or protoplasm, what will happen? It will be broken. So glucose is 6 carbon compound. this glucose will be 6 carbon compound. It will be broken down into 2 molecule of 3 carbon compound. That is a pyruvate. The process of breaking a glucose means 6 carbon compound into 2 molecule of 3 carbon compound, that is name is pyruvate. This process is called glycolysis. So this complete glycolysis process goes on in cytoplasm. During this process, formation of 8 ATP will take place. In the nut cell, 8 ATP will form. So is that clear?
Yes, sir.
Inside cytoplasm, there is mitochondria. This is mitochondria, right? Pyruvate can enter. Why glycolysis is needed? You can ask me why not glucose itself goes inside the mitochondria. Mitochondrial membrane do not allow the entry of the glucose. They cannot enter inside the inner membrane of glucose. They cannot cross the inner membrane of this mitochondria. That's why it breaks down into pyruvate, right? Please note down. Glucose can enter. Glucose can enter in the cell. Enter in the cell through glycolysis. Sorry, can enter into the cell in plasma through plasma membrane. Through plasma membrane. Full stop. Glucose cannot cross the inner membrane of glucose cannot cross the inner membrane of mitochondria. That's why it breaks down into a 3 carbon compound. That's why it breaks down into a 3 carbon compound pyruvate. Let's stop. Pyruvate can go inside mitochondria. Pyruvate can go inside the mitochondria and reaches to matrix. Now pyruvate cannot participate into Krebs cycle. So the complete burning takes place in Krebs cycle. To participate in Krebs cycle, it is further broken down into 2 carbon compound acetyl coenzyme. Now this acetyl coenzyme will be this is acetyl coenzyme. Acetyl coenzyme will be oxidized or broken down into Krebs cycle in the matrix. Carbon dioxide and energy will be released. Energy will be released in the form of ATP, NADPH, and FADH, right? And this is equivalent to 38. So 8 ATP are produced in glycolysis, 30 ATP in Krebs cycle. So that's how from one glucose molecule, total 38 ATP molecule are produced. Is that clear or not? Tell me.
Yes, sir.
So Rabia, GTP is equal to ATP. That you need to remember. 1 NADH is equal to 3 ATP. 2 FADH is equal to 2 ATP. Right? That also you need to understand. Okay. So this was the complete summary of the glycolysis. How does the glycolysis take place? Now the glycolysis. Look, what you need to remember or what you need to cram, Rabia, whatever is underlined with the red, that you have to cram. There is no, and you have to cram this process. There is no other process. So how you will cram this, Rabia? Any idea? Can you give me the idea to cram it?
Like To go through the topic and the terms I can understand.
Hardly will go one time, two time, or three times. So better, wherever is your study table, make this flow chart in A4 sheet, even in a smaller sheet if it's possible for you. So make a A4 sheet, right? And this A4 sheet, paste it in front of your or your chair, or in front of your this one, the study table, right? At the wall, front of your wall, at the study table, and just go through with it as many times you can. So you will see, without any intentional cramming, unintentionally you have learned it, right? And this is it's a very old technique, but it's still working. It's still a good technique. You can do, right? And that is the best way, as per my view, right? Look, in glycolysis, first, this glycolysis is also known as the EMP pathway, right? So glycolysis is also known as the EMP pathway. The reason behind, there were three scientists who discovered this: Emden, Meyerhoff, and Parans. So the three scientists were Gustav Emden, Otto Meyerhoff, and J. Parans. So all these three discovered after them, we call them EMP pathway. So glycolysis is also known as the EMP pathway: Emden, Meyerhoff, Parans pathway. Uh, we divide glycolysis into two parts: preparatory phase and payoff. What is the preparatory phase? In preparatory phase, it consumes the energy, right? It consumes the energy. It requires the energy. So it consumes the energy, and after that, so from here till this point, this is preparatory, and from here onward, from here to here, this is the payoff phase. In payoff phase, it will give you back energy. So try to understand. Glucose will break down into glucose 6-phosphate. ATP is spent. So I have written over here minus 180. ATP will spend over. Glucose 6-phosphate will break down into fructose 6-phosphate. In presence of, it will take place in presence of isomerism. Both are the isomer of each other. Glucose 6-phosphate will convert into fructose 6-phosphate. Again, the fructose 6-phosphate will break down into fructose 1,6-biphosphate, and again 1 ATP is required. So from glucose to fructose 1,6-phosphate, total 2 ATP are consumed. That's why it is called preparatory phase. Here preparation is taking. Now this fructose 1,6-biphosphate, it will break down into two substances: G3P and DHAP, dihydroxyacetone phosphate, and GTP, right? Now this GTP will participate in phase cycle, not DHAP. So this DHAP will convert again into G3P, glyceraldehyde 3-phosphate. G3P will break down into 1,3-biphosphoglycerate. When J3P will break down into 1,3-biphosphoglycerate, formation of 2 NADH will take place. 1 NADH is equal to 3 ATP. I have already told you. So formation of 2 NADH will take place. Again, it means the production of energy is taking place. Again, 1,3-biphosphoglycerate will break down into phosphoglycerate. Again, formation of 2 ATP will take place. 3-phosphoglycerate will convert into 2-phosphoglycerate. Then further 2-phosphoglycerate will convert into phosphoenol pyruvate, PEP, and PEP will convert into pyruvate. And again, the formation of 2 ATP will take place. So here we can see how many ATP are produced. So minus 2 ATP are consumed, minus 1, minus 2 ATP are consumed, plus 2 NADH are produced, 2 ATP are produced, again 2 ATP are produced. Look. So minus 2 ATP, I have again written over here, minus 2 ATP. Again, 2 NADH is equal to how many ATP? So I have already said to you that 1 NADH is equal to 1 NADH is equal to 3 ATP. So here 3 into 2. So here it will be total 6 ATP plus 2 ATP plus 2 ATP. So how many ATP? So these are total 10 ATP. From 10, 2 ATP are consumed. So our net gain is 80. That you need to remember what is the net gain of So minus 2 ATP are consumed in preparatory phase and 8 ATP are produced here. Go through with it and tell me, is it clear or not? And if it's clear, you have to note down the energy balance. That's very important. This is called energy balance. Tell me, Rabia.
Yes, sir, it's clear.
All clear?
Yes, sir.
So please note down the energy balance sheet. This one. These both two balance. That you need to note down.
So,
Yes.
what is that word above +2 NEDH?
Sorry. Say again.
What is that word above +2 NADH?
Plus 2 NADH. This one. It look here when glyceraldehyde 3-phosphate will break down to 1,3-biphosphate, right? So 2 NADH are produced. Clear?
Yes, sir, but what is above that? Like -2?
This one. Okay. Minus 2 ATP are consumed. This one. Okay. Let me write it down more clearly. Minus 2 ATP.
Done, sir.
Done. Okay.
Yes, sir.
So this was the glycolysis. And what you have to remember after the class, you have to remember only those things, whatever I have underlined. Now come to the Krebs cycle or TCA cycle, tricarboxylic acid cycle. So the compounds have the tricarboxylic acid. That's why we call it TCA, tricarboxylic acid cycle. And it is also known as the Krebs cycle. Look. Now, glucose is a 6-carbon compound. Try to understand. Glucose is a 6-carbon compound. It will break down into pyruvate, 2 molecule of pyruvate. And pyruvate is 3-carbon compound. So 2 3 are 6. Again, there are total 6 carbons. Now, what will happen? I have already said that pyruvate cannot enter in the Krebs. Pyruvate will break down into acetyl coenzyme A. When pyruvate will break down into acetyl coenzyme A, one carbon dioxide will be released, and this is called decarboxylation, right? This is called decarboxylation. Again, formation of NADH will take place. Means this is production of energy is taking place. So formation of NADH2 will take place. Acetyl coenzyme A will convert into citrate. Citrate is 6-carbon compound. So try to understand. Here 2-carbon compound, this one from here, and 4-carbon compound of oxaloacetate, right? So they are this acetyl coenzyme A and oxaloacetate, they will combine and they will form citrate. Citrate is 6-carbon compound, right? So 2-carbon compound from acetyl coenzyme A and 4-carbon compound from oxaloacetate, right? So they will combine and they will form a substance citrate, that is 6-carbon. Citrate will convert into isocitrate, again 6-carbon compound. Now the citrate, isocitrate will break down into alpha ketoglutarate, a 5-carbon compound. Again, the one where is the 1 carbon lost? In the form of carbon dioxide. So again, the decarboxylation taking place. 1 carbon dioxide will be lost, and again the production of NADH will take place. Means this is the second time where energy production is taking place, right? Alpha ketoglutarate will break down into succinyl coenzyme, which is 4-carbon compound. Again, 1 carbon will be lost in the form of carbon dioxide. Again, production of NADH will take place. This is third NADH is produced. Now, succinyl coenzyme, a 4-carbon compound, it will break down into succinate, a 4-carbon compound. Formation of ATP will take place from GT, right? Formation of GTP will take place from GTP. It means GTP is equal to ATP. I have already told you, right? Succinate will break down again. Oh. Succinate, a 4-carbon compound, will break down to fumarate. Look, the carbon dioxide is not evolved, but formation of FADH2 will take place. 1 FADH2 is equal to 2 ATP. Fumarate will convert into malate, a 4-carbon compound. When malate will convert into oxaloacetate, a 4-carbon compound, again production of NADH will take place, right? Again, oxaloacetate will combine with oxaloacetate, will make. So this is complete cycle is called Krebs. Now try to understand how many NADH are produced. So try to understand. So how many total NADH are produced? Let me count. 1. Try to understand. 1, 2, 3, 4, 3, and this is 4 NADH, right? 4. So there are total 4 NADH at 4 places. So no. 2 NADH will be produced at every point. It should be 10. 1 point where the NADH 1.1, and this is second point. This is third point. This is 4 point. There is one more point. The total, that means glucose 4 ADP, respiration 10 NADH, 2 FAD. Oh yes, 8 in Krebs cycle. So in ATP we have understood total. Sorry, in glycolysis total 8 ATP are produced. Yes, in 4 point. So total, look, at 4 point NADH are produced. 1, 2, 3, and this is the 4th point where NADH is produced, right? So every time 2 NADH are produced. So 2 4 ja 8 NADH are produced. 8 NADH are here. 8 NADH is equal to 24. 8 3 ja 24, right? Again, where is the FADH2 is produced? So at one point FADH is produced. This one. So total 2 FADH are produced. So try to understand. 2 FADH. So 2 2s are 4. So total how many? 28 ATP are produced. Now this 2 GTP are produced here. 2 GTP are produced. So GTP is equal to ATP. So 2 ATP are produced. So in Krebs cycle total 30 ATP are produced. In glycolysis total 8 ATP are produced. So 30 plus 8, total 38 ATP are produced during the process of glycolysis. Right. That is the balance sheet of the. This is called respiratory balance. You can note down this reaction and you have to note down this chart, this respiratory balance. Because the multiple time the question have been asked from this. And tell me, is it clear or not? If you have any doubt, please ask me right now from this cycle. If you have any doubt, please ask me from this cycle. Then you have to note it down.
So can you explain what is malate dehydrogenase?
Malate 2. This one, okay. This malate will break down into oxaloacetate. right? Malate is 4-carbon compound. Again, oxaloacetate is 4-carbon compound. When malate will convert into oxaloacetate, formation of NADH will take place. Again, this oxaloacetate is 4-carbon compound. This oxaloacetate will combine with acetyl coenzyme. Acetyl coenzyme is 2-carbon compound. So malate plus oxaloacetate plus acetyl coenzyme means 4-carbon plus 2-carbon is equal to 6-carbon. So combination of acetyl coenzyme and oxaloacetate, they will form a 6-carbon compound that is called citrate. Tell me, is it clear?
Yes, sir.
Done? Is it clear? Is this cycle is clear?
Yes, sir.
Then you have to please note down this energy balance. Copy this because this is important for your exam. And you have to remember it. That's why I have jotted down all the point at the same PPT.
Sir, there's a part like the equation will be asked.
This equation,
Like,
this equation. No,
yes,
you need
sir.
not. No, you need not to cram this equation. You have to just go through with this one. First process glycolysis. How many produce? Second Krebs cycle. How many ATP are produced? And total 38. You know, one more point. Many of the time the question have been asked how many ATP are produced during the Krebs cycle or oxidation, complete oxidation of glucose. Sometime you will get option 36 and sometime you will get as an answer 36, but that 36 is wrong. Even if it is written in the many of the stalled question bank, I have seen the 36 is answered, but 36 is not correct. That is 36 is wrong answer.
Done, sir.
Done.
Yes, sir.
Electron transport chain will study next class because it will take some time. Like how the production chemospheric fuel, amphipholic coffee. Almost this is done, but only the thing which you need to cover, which you need, that is the electron transport chain, right? And let's do this thing, anaerobic respiration. In the meantime, because we have 4 minutes or 5. Look, in anaerobic respiration, this is anaerobic pathway basically, right? So glucose breaks down into look, either it's a one more thing I want to tell you. Either it's aerobic respiration or anaerobic respiration. Glycolysis will go on. That is same for both of them. So glucose will break down into G3P, glyceraldehyde 3-phosphate, right? G3P will break down to 3-phosphoglycerate, PK. Then it's phosphoenol pyruvate, PEP. PEP will convert into pyruvate. This is complete process of glycolysis from this point to this point. This is glycolysis. It will go on in the anaerobic respiration. Here, from pyruvic acid onward, in aerobic respiration, Krebs cycle goes on. Here, Krebs cycle do not go on. So, here pyruvic acid will convert into lactic acid in human muscle, right? And formation of NADH will take place, right? Fine. So, NADH will be produced, and in the bacteria, they do not require oxygen. Fermentation is anaerobic respiration, right? In human muscles, what will happen? This for formation of lactic acid will. And in bacteria, fungi, and all that, pyruvic acid will break down into ethanol and carbon dioxide. Again, the consumption of NADH will take place. NADH will be brought down into NAD, right? Ethyl alcohol is produced in with the help of yeast. So pyruvic acid will convert into acetaldehyde and carbon dioxide. Acetaldehyde will convert into ethyl alcohol and NAD plus, right? This is for the yeast. In bacterial fermentation, so this process is called fermentation, means the process of production of ethanol or ethyl alcohol in the anaerobic respiration is called fermentation. Fermentation can be two types: yeast fermentation and the second is bacterial fermentation. In yeast fermentation, what will happen? Pyruvic acid will break down into acetaldehyde and carbon dioxide. Acetaldehyde will break down into ethyl alcohol and NAD. In bacterial fermentation, the formation of lactic acid will take place. Pyruvic acid plus NADH, they will make lactic acid and NAD plus, right? So this is the process of fermentation, and this is called anaerobic respiration, right? So this was, go through with it and keep it in mind. So you have to focus in the bacterial fermentation and yeast fermentation. What is the difference? Is that clear, Rabia?
Yes, sir.
Okay. So the only thing which is left in this chapter, that is an electron transport chain. We'll do in next class, right? And see you next class. In the meantime, what you have to do, you have to revise all these parts, right? You have to all this concept, you have to revise all these concepts. You have to take care of those parts where I have pointed out, fine. And in case if you get any question in your NCRT, in case if you get any question in your question bank, please note it down and discuss with me next, right?